So the lecture started machine learning for precision medicine avenues and roadblocks. Now, what I have to immediately say is that our research is focusing on neuroimaging, in particular MR imaging, as a tool to measure biological information in the brain. And then, obviously, when we talk about precision medicine, the focus will be on brain medicine. That being said, I sincerely think that virtually all of the points I'm raising here will be highly relevant for any sort of data in the context of biomedical applications. And in fact, we do have a lot of collaborations with different medical specialties, from internal medicine to pediatrics, surgery. And we really see that these are not domain-specific issues, but the fundamental aspects that I will outline in this talk, they really hold across the entire field of biomedical applications. So let's start. We all know that humans are different from each other. If we would be in a lecture hall, I would ask you to look left, look right, and you immediately realize that we differ from each other in many aspects: age, gender, height, hair color, skin color, whatsoever. And likewise, we also differ in our experiences, in our health, our lifestyle, and so on. And hence, it's not surprising that we do not only differ from each other in terms of externally visible physical phenotypes, but there's also a marked inter-individual variability in virtually any measure about neuroanatomy. So when we look at brain structure, function, or connectivity, we always see a substantial variance between individuals. Last but not least, humans also differ markedly from each other in terms of what I would call inner traits. So something like cognitive performances, working memory, attention, socioaffective functions, your personality, your mood, and last but not least, obviously also our predisposition for or our suffering from brain disorders. That means psychopathology. Now, the big question is, can we put this together and can we link the variability in what we would consider driving factors, such as age and gender, to the variability of the endophenotype, which is neuroimaging data, and the variability in observable psychological or clinical phenotypes. And with that goal, we very, very quickly come to the end of what's called the classical biomarkers. Because while there is a very rich literature on biomarkers, I mean, just do a PubMed search, you find millions of hits. Virtually all of them actually represent within sample comparisons or associations. So basically, the entire body of biomarker literature really just asks, is there a surprising difference in the mean of the patients for a given feature, for a given measure, and the mean of the controls for the same measure? And I show you an example here. And this is a figure, as you would see millions of times when you look at the PubMed. So you have patients, you have controls, you have a highly significant difference. And if you look at the standard errors, they are very well separated from each other. And this brings me over to the critical point that while this describes something that seems to be different in patients than in controls on the group level, we really have to question whether this is an actionable marker for clinical practice. And let me show you the exact same data, the exact data I used for these plots, just in a somewhat more honest fashion. And as you can see here, despite the substantial between group differences, so even though the group means were highly significantly different from each other, there is a vast overlap in the individual expressions, both among patients and controls. So this means that a biomarker, a classical associative biomarker, may be useful to better understand a disease, but it is usually useless when we want to make individual statements. Critically, though, clinical statements are always about the individual. If I'm a neurologist and I see a patient, I do not want to know something about Parkinson's in general. I want to know something about that particular person that is sitting in front of me. So we have to change our perspective from the classical within sample comparisons to learning patterns in the data that generalize to new cases. And this is obviously where we are now in the field of machine learning. So rather than testing whether our patients are significantly different from our controls, we want to train a model that learns the multivariate relationship between the features, the things that I have that I want to predict from, and the target, the outcome that I do want to predict. And then I have to test that model in a new independent test group and see whether the learned associations actually generalize, allow a correct prediction of the target in new subjects. Now, in the field of brain imaging, and again, I do really want to generalize that in the field of biomedical research, we are entering a era where we now have the chance to actually do something like this. We have very large cohorts. We have the infrastructure in terms of data management and high throughput computing, and we have sufficient a priori knowledge on the domain of interest due to the biomedical literature over the last century, really, that we can start to build machine learning models for individual assessment. So, full steam ahead. And in some way, this indeed has been the overarching theme of our field in the last 5 to 10 years. I'll just give you a very obvious example for modeling interdivisional variability. What you see here is a brain scan, and here's a second brain scan. And I think everybody, even though maybe you're not from a neuroscience background, you can appreciate that the right image, there seems to be quite a bit missing. So where on the left side, you do have the brain, the gray and white. Then on the right side, you have black, which is cerebrospinal fluid. I mean, this is atrophy. This is degeneration. And I will kind of shock you, though, that what I show you here is not an image of a patient with Alzheimer or another dementia, but rather the scan on the left is from a 20-something-year-old. The scan on the right is from a person that's in their 60s. So what we're looking here is the normal aging process. Now, aging is a highly individual process. If you look at people in their 50s, 60s, and 70s, you see a vast divergence on their cognitive and motor skills. And likewise, we see a large divergence in terms of neurobiology. And so the question you can now use machine learning for is to ask in spite of all of this inter-individual variability, are there canonical patterns? Can we model this aspect, aging, of inter-individual variability such that for a new subject, just seeing the brain scan, we can estimate how old that person is? And the quick answer is yes, we can do so very well. So in healthy subjects, we have a precision of plus minus 4 years. And we also see that the divergence between biological and predicted age and chronological age is related to general health, lifestyle, and cognition. So what this means is that we can use machine learning to develop canonical models of variability, and we can then assess patients with different, in this case, neurological and psychiatric disorders against this kind of canonical model. And as you can see here, we do see a, in some cases, very substantial advancement of individual aging in patients with neurodegenerative diseases. If you look at the bottom, Alzheimer and multiple sclerosis, their patients on average look about 10 years older. So somebody who's in their 60s has a brain that looks so that the model thinks that person should be in the 70s. Here's a more concrete example on Parkinson's, and I want to extend that, what I just said, to yes, there's also a relationship to individual symptomatology, individual, in this case, motor symptoms. But maybe more importantly, we can also use machine learning frameworks to actually classify patients from healthy controls. So that's what you see on the right side. Here we have the perspective specifically on the individual, asking if we train a model on a larger group of patients and controls, can we correctly predict, can we correctly assign a new subject as being either a patient or control? And as you can see, we can do this with a fairly good accuracy, about 83%. Now, you could argue these are all things that a good radiologist can do anyways. So any radiology report has a statement on looks pre-aged, looks like a Parkinson pattern. But can we also see things that even a very skilled radiologist cannot pick up? So this is the example here I just showed you on young versus old. Here's a different example. Now, let me assure you that the top and the bottom, these are two different people, two different persons. And since they're about the same age, you can't really spot any differences. Now, let me reveal what's different between them. The top image is from a male subject, the bottom from a female subject. And we just had a bit of fun with the neuroradiologists in our area and tried to see how well somebody who has seen thousands and tens of thousands of scans in their training and professional work, how well they could differentiate male and female. And the answer is not at all. So even very experienced neuroradiologists are absolutely at chance level. If we do this, however, in an algorithmic fashion, so we trained a model on about 1,300 subjects and tested in about 300 new cases, you see that it's actually not so difficult to get a very clear differentiation. Now, what does this tell us? First of all, it shows that we can model these two broadest domains of inter-individual variability, aging and sex, quite well using machine learning. But what to me is really critical on this example is that we can find patterns in the data that are, for all practical purposes, non-accessible to human readers. So we can get good generalization results, even on aspects that cannot be differentiated by experienced neuroradiologists. And from there on, we can obviously move on and see whether we can also predict something that's not even yet happening. So this is a study we did together with colleagues in Aachen on tinnitus. So tinnitus is this very annoying sound, phantom sound in the ears. And they were evaluating a new approach to treatment used based on transcranial magnetic stimulation. And what they found is that it's basically about a third of the patient that respond to treatment, whereas the majority does not. And then we had a look at the scans that were taken prior to any treatment and see whether these allowed to predict who will benefit or not. And in the end, we achieved an accuracy of about 85%. So what you see here on the right is, as I just said, most of the patients do not benefit. So the blue bars are larger than the red ones, but we get a good accuracy in classifying who will, in the future, because the treatment has not started yet, benefit from the treatment or not. So overall, and combining more evidence that we can also predict individual cognitive or behavioral phenotypes, we are in what feels like an extremely good spot. So using objective markers, such as brain imaging, we can predict individual clinical behavioral phenotypes. We can model major aspects of inter-individual variability. We are in a place that feels like, as I mentioned in the beginning, full steam ahead. But unfortunately, there's the second part of my talk. And that second part of my talk will now become a lot more critical, because what I think is really important to realize is that while we have extremely promising results, while it feels like we have the avenue towards objective, reliable, and valid precision medicine based on machine learning approaches, there are quite a couple of bumps, maybe even blocks in the road that I feel need to be addressed before we can confidently actually move into the public domain and tell the wider population that we are now on the next level, brain medicine 2.0. So let's have a somewhat closer look at the overall machine learning pathway and some of the bumps and blocks that we will encounter on that road. Obviously, we will start with the data, the features. That is what we want to predict from, and our targets. That is what we want to predict. Now, in virtually all cases, and again, I'm focusing on neuroimaging, but this is true for any sort of biomedical information, you usually cannot use the raw data as input into your algorithms, but the data needs to be represented or processed or both into some form of features. Then features and target get fed into the model, are used to train that model. Then we validate that model, and finally we come to the deployment of a product. Moving along that chain, I just want to highlight a few aspects that hopefully illustrate that while the overall pathway is clear and easy, the details, if you look closer, get more muddy. And here's a very straightforward example. So here we have the T1 structural MRI of about 2,200 subjects. And what we did is just to process them from the raw image to the volumetric representation. That's what you see on the top left here. The left part is the raw image. and the right is the amount of gray matter, the amount of brain tissue at each location. And there are basically a couple of standard tools. And with standard tools, I mean out of the box. I don't go into the details here on the imaging side, but it's rather important that these are the kind of models that you can download, the tools you just install that have been used thousands and thousands of times before, and you would never question their validity. What is a bit interesting, though, is that when we tested 10 BBM pipelines, which is basically the standard models with some combinations of their different components, we actually saw something really surprising. Namely, there was only a low to moderate agreement between the different processing workflows. Now, what do you see here? What you see is basically for 400 brain regions, the correlations of the individual volume estimates across algorithm. So basically, in an area that is dark red, the individually estimated brain volume is the same no matter which of the standards pipelines you use, or they're at least very highly correlated. If you then go, let's say, to the bright yellow, though, these are regions where the individual per subject volumes are virtually uncorrelated between different pipelines. What's the take-home message here, particularly for those who do not use imaging? The take-home message is that it starts at the very bottom. So I like kind of the provocant question here. What, if anything, is my data? I mean, trust your data. Let the data speak for itself. Are we really sure what is our data? Because we very rarely use the rawest form of the data, be it imagings, be it recordings, but we usually have some form of processing put on top of it. And this can already make quite some difference, even though in common practice, we would refer to the output of the initial processing steps as our data. But you should remember, this is not your data. It's already some combination of data and approaches. Next aspect, you need to do some form of data representation, which means that usually your data is too big per subject and you don't have enough subjects. This is a very peculiar aspect that everybody who works with biomedical data knows, but seems to be very surprising to those people who do not work in biomedicine. The key point here is that our elements, our number of observations is really the number of patients, the number of subjects. So it's easy to acquire more data per subject. But your key degree of variability is really represented by the number of samples you have. And the number of samples is usually small, way smaller than you would want, because these are the distinct individuals, the distinct patients you can measure. So this quickly leads to a situation where you have very unfortunate feature to sample ratios. So in the case of brain imaging, we easily have 10,000s, 100,000s of measures because that's our individual image points. But usually we are limited to a few hundred, maybe a few thousand subjects. So we need to do some form of data representation, in our case, often atlas-based compression, and there's a lot of different atlases you can use. And what is illustrated here, the way you represent your data has a substantial influence. I put that together and then come to an extension of this thought. You have originally measured something. That's the raw measurement. You usually do some form of processing to get the process measures, and then you do some form of representation that actually gives you the features. Both of these steps have a profound influence on the actual features that you get out at the end, the things you feed into your machine learning models. Now, I'm happy to anticipate that some of you will argue, well, that's what we have deep learning for. With deep learning, you don't need processing, you don't need feature representation. You just put in the raw data and it's end to end. Yes, but no. Why is this, unfortunately, an illusion, a fallacy? Because basically, if you use deep learning, you're just replacing processing and representation with model architecture, network architecture. So you don't need to make the choices of which software you use for original processing. You don't need to make choices of how to represent your feature. Well, at least you don't need to do it consciously, but in fact, you are doing it all the time when you set up your model architecture. How many layers, what kind of convolutions, what kind of relay units, what you're adding here and there. It's all the same for deep learning. It's just named differently. And when we come to the next aspect that I want to highlight, it's actually identical. And this is the target. We kind of underappreciate the effect that our targets have on our models as well. Now, let me explain that. Here we have done some age prediction, as I showed previously, and we see that with R2 as an accuracy metric, we can get a quite nice prediction. Now, what we've done next is to use the very same pipeline, but artificially degraded the reliability of the target, basically adding some measurement noise to the target. What do we see? Accuracy drops massively. Well, this is kind of artificial. And does it only hold for age? No, it also holds for all sorts of cognitive phenotypes, cognitive capacity, crystallized cognition, grip strength, total cognition. And we also see a strong correlation, very strong correlation between the experimental reliability of phenotypes. So this is really measuring test, retest reliability of the phenotypes and our accuracy in predicting them. And this, which also holds in other databases, has some profound impact on machine learning models. If you have a reliable phenotype, if you have a reliable target, then basically we always see this curve. You increase training set size, you get a better accuracy. And then at some point it will saturate. That's basically the overall capacity of how good you can predict the particular target. Straightforward. More data, better predictions. With a good reliability. Now, if we degrade reliability, what you see here is that this effect is a lot stronger. And in fact, if you have an unreliable target, then no sample size can save you. At some point, it's just impossible to predict anything meaningful. And again, this is not just a theoretical consideration, but for anybody working with biomedical data, in particular working with behavioral or clinical data, this is probably the major problem that holds back your work, that holds back the entire field. If we cannot construct reliable targets, and unfortunately, diagnoses and clinical measures are notoriously unreliable targets, then we cannot just resort to bigger data, better algorithm. We will hit a roof quite quickly that is determined by our targets and that we cannot break with just working on the data science side. So what's the overarching message here? I already said, don't trust your data. What, if anything, is your data? I already said that you need to be aware of any transformation and representation, be it explicit or be it through the model architecture and deep learning, has a big influence on your prediction pipelines. But on top of that, there is a intrinsic limit of how good you can predict any particular phenotype, any particular clinical or medical target that is determined by the reliability of that measure. So conversely, that means you can optimize here on the left side all you want. You can spend years on algorithm optimization. If the target is not reliable, no data science will help you. And this is a somewhat uncomfortable realization, but I think it's a very important one, in particular when working in the biomedical field. Now, let's put things together. We have our features, whatever that is. We have our target, however good that is. And then we run our model. And there are some cases, as I showed earlier, in terms of age prediction, where it seems that prediction is really a very robust, very straightforward aspect. Now, what Shamimura, a former PhD student, has done is basically just play around with this prediction workflow. Keep basically the targets and the data, but just use slightly different settings, different algorithms, and so on. And again, similar to what I said about processing, this is all the standard out of the box. This is the standard. You see dozens or hundreds of papers doing all of that. And what we clearly saw is that the more or less at random methodological choices result in a substantial variance of algorithm performance. So this is why I've become a very big fan of multiverse analysis. We do not see this very often. But basically, from my perspective, any paper that shows a nice prediction in the biomedical field only based on one particular algorithm, on one particular workflow, you should not trust it. Because it could be just either by design or at random hitting a good spot in these combinations. So you really would encourage some form of a multiverse analysis where you show that your prediction model is not dependent on the exact settings. And again, that holds for classical machine learning as well as deep learning, where in deep learning, you just need to replace data processing, feature representation, and to some degree model with architecture. But there's also some other part that is more intrinsic in the meta, in the domain. And this is illustrated here. So we try to estimate cognitive processing speed from MR imaging data. We used a lot of different algorithms, a lot of different measures. And what you can see quite nicely between 50 and 20,000 subjects, there's more variance down here. But on the right side, we get a fairly good structure. So basically, some approaches work better than others. But in general, more data helps better predictions. Well, this is all nice. This is definitely a step forward from the standard paper in the field that just tests one algorithm. But then we also did this. Here we predicted the same target just using confounds, namely age, sex, and intracranial volume. And what we saw is that throughout the entire range of data sizes, our confounds always did a much better job in predicting the target than any of the exquisite and highly advanced features and measures that we derive. And this is not a singular observation. In fact, when we looked at the gender prediction, we actually saw the very same picture. What the algorithm actually learned is that men are on average taller than women, and hence it was using head size as a shortcut for sex classification. So where does that lead us? It basically comes down to the aspect that human variability is likely rather low dimensional. So in large biomedical data sets, we can measure dozens, hundreds, thousands of information for everybody. If you look at things like the UK Biobank, there's, I think, 10 or 20,000 data fields per subject. But we, the people, we as people, we do not differ from each other in hundreds of thousands of independent dimensions. Rather, inter-individual human variability is probably low dimensional, as in like maybe a few dozen broad dimensions that then shape a lot of different features that we can measure. So with that understanding, we really have to challenge the traditional view of features of interest versus confounds and rather consider confounds as somewhat of a gradient. I mean, obviously, there are avoidable issues, such as bad samplings. All your patients are male, all your controls are female. That's kind of like really an avoidable confound. But then there are things that you would consider have an implausible, yet, but it may be biological link. So one of my favorite examples here is that there are GWAS, genetic hits, for the question of how often do you eat bacon for breakfast. It's highly significant. Well, maybe that has something to do with your saline receptors in the tongue. Maybe that has something to do with sociocultural background. Then there are other aspects that we can quite easily think as reflections of latent underlying dimensions. Lots and lots of things, virtually anything, is related to age and gender or sex. So basically, there are some very broad latent dimensions. We get more specific, we often have different data points, different features that are driven by common factor. And last but not least, we have things that basically measure the same biology just with different instruments. Where does that lead to? It basically leads to a two-dimensional perspective on confounds. One is the what we call statistical perspective. In large biomedical data sets, really everything is correlated with everything. So here is a simple outcome, hand grip strength, which we use because it has a very high reliability. And basically, tons of different measures from age to and sex to things like blood pressure and length of the working week are correlated with hand grip strengths. But then also there is the more conceptual dimension of confounding, where we do have a gradient from naturally biological links, basically biased convenience samples, to, in our case of hand grip strengths, aspects such as sex, where we have a very clear biological pathway from sex to testosterone production to muscle growth to hand grip strengths. Where does all of that end up? Basically in a two-dimensional on the matrix where we have to consider both the statistical and the conceptual relation of a confound, and we cannot get away with the traditional perspective of simply regressing out a few standard things or looking at how strong something is related. Again, intermediate fazit, your data is outside of the very raw data, a somewhat flickery construct because it depends on what you do with it, and even more, how do you represent it as features in the machine learning model? Your target, the thing you want to predict, has a substantial input, particularly through its reliability. And when it gets to the actual modeling part, we really have to be very considerate and spend a lot of domain aware thoughts on what we would consider a confound, a unwanted shortcut for the model, or not, i.e., something that is a true relationship that we cannot remove from the data because it actually is the same biology. Once we've been through all of that, we obviously need to validate our models. And this figure here on the right now illustrates something that should be fairly well known, but is often somewhat ignored, and that is the relevance of batch effects in biomedical data. And if you look at human data, human subjects, i.e. patients, not just cells, for example, then we do have actually a quite strong exaggeration of batch effects, because not only you have the same technical batch effects as you have from two sequencing machines, but also the samples that are recruited, let's say in Düsseldorf versus Munich, the samples themselves may be different in their composition, age, socioeconomic background, cultural history, and that exaggerates batch effects quite strongly in biomedical patient-centered research. And hence it's not surprising if you look at this, you have a quite good performance along the Mine diagonal. So you train on a particular set and test on the same set, things are quite okay. If you set on a test on a completely independent set, then things look much bleaker. So batch effects are critical, but you can actually, if you are aware of them and if you explicitly want to address them, then you can get away with it, for example, by training on a mixed data set that is made up across data from all different training sets that generalizes good to all of the original sites, plus also the new test set. And what's quite interesting in some cases, here, virtually all cases actually, the data trained on the mixed set outperforms the main diagonal. So injecting more variability, perturbing the data more, may actually be beneficial also for the within set generalization. Why is that really, really critical? Well, I just mentioned that there may be aspects that differ between samples. And here, Jingwei Li actually looked at this very explicitly by basically looking at a sample of white versus African Americans. And what we saw, and I just really summarized that briefly, is that models trained on the majority group can do all sorts of bad things on the minority group. And with all sorts of, I mean, really all sorts of, you get worse predictions, you get baseline shifts, biases, you get gradient shifts, you get a completely unpredictable bunch of problematic behaviors by the algorithm. And this is obviously critical when we finally think on the next frontier, which is the actual deployment. In a system such as dimensional variability, there will always be tons of shortcuts available to the algorithm because everything is interconnected. And there are these big, I would call them overarching tides or waves, such as age, gender, ethnicity, that influence a lot of things. And if we are not careful of how to model that, we extremely quickly end up with algorithms that are fundamentally biased and ultimately unfair in the sense that they do uh, base their predictions to a quite large degree on confounding aspects. Just take the example I just showed about male-female. Some female that is very tall, has a large head, will be more likely recognized as a man, whereas somebody like me, who is not the tallest in the world at all, just by head size would quickly be assumed to be female. And that may have substantial influences on subsequent medical decisions. And it's even worse with things like ethnicity, social economic status, and so on. So when we want to move into the domain of actual real life applications, I think we all need to realize we are leaving a sandbox here. We're leaving the abstract world of R is a bit larger, is a bit smaller. Here we actually come to a scenario where acceptance on two levels by the patients and by the doctors, and ultimately also the responsibility question becomes extremely relevant. And in the interest of time, I will not go into the details. I mean, we've written about this quite a bit in the past, and I think Bert Heinrichs at some point also talk about this. But the bottom line is all of the problems I noted, data, feature, representation, targets, models, confounds, and whatnot, they all influence your final product. And it's very, very likely and dangerous to assume that you can just deploy in the broader population, not knowing how major aspects such as sex, gender, ethnicity, and socioeconomic status interact with your models and their performance. So to wrap up, where do we stand? Full steam ahead or into the thunderstorm? I think it's kind of both. I mean, we do have the unique situation that now, for the first time, machine learning allows an objective, individual prediction of health and disease. But the performance of the complex workflows we need for this can be quite a bit more brittle than we want. And when it comes to biomedical applications, conceptual and biological challenges meet ethical, legal, and societal ones. And with that, I would like to thank all the people that have been involved in the different parts of this work. Obviously, I want to thank our funders. Thank you for your attention, and I'm now very happy to take questions. Thanks a lot.